Hi everyone, my name is Laura Court and I am a medical device guru here at Greenlight Guru. This session presented by Audrey Turley, a senior biocompatibility expert at Nelson Labs, is on biocompatibility evaluation for device submission. As always, you're encouraged to ask questions in the box on your right-hand side and we will get to as many as time permits. Attendance certificates for all of our sessions are offered on Greenlight Guru Academy at academy.greenlight.guru. And if you're looking for further education and training, you may be interested in our webinar on evaluating need for biocompatibility testing and mitigating risk for changing your medical device. This webinar was presented by our partners at Nelson Labs at a previous summit series, but is still available for free on Greenlight Guru Academy. Be sure to check it out. Finally, if you're curious about Greenlight Guru and how our software can help you get to market faster with less risk, be sure to head over to our website and request a free demo after this session. Now to give Audrey Turley a proper introduction. Audrey has over 25 years of experience working in research, laboratory, and test design functions in the medical device industry. She is a biocompatibility expert, having spent 15 years in the lab performing all of the in vitro tests offered at Nelson Labs. Audrey was responsible for the development of the in vitro genotoxicity assay at Nelson Labs. She was also on the team at Nelson Labs, which developed and validated the in vitro irritation assay for medical devices. Audrey is an active committee member for several working groups with the Association for the Advancement of Medical Instrumentation, AAMI, and on the International Standards Organization, ISO level as well. Audrey currently leads the team of biocompatibility expert consultants at Nelson Labs. With further ado, I'll hand it over to you, Audrey.
Thank you so much, Laura, and thank you everyone for joining us today. I just want to caveat my presentation right up front saying that I've been given 30 minutes for a presentation and I can talk about biocompatibility for 3 days. So we're going to pack a lot in today. So I just encourage you to ask your questions and just realize I'm going to give you an overall approach and some thought processes that we've used successfully before. And so a lot of this will just be guidance to help you think about your own specific situation. And so I think our first slide, Laura, is one for Greenlight Guru to speak on. Is that correct?
Yes, so this is more just a quick summary of Greenlight Guru and all of the great different features that we have as well. If you would like a demo on our software, our Greenlight software, to help you get to market faster and create a true quality medical device, you can head over to greenlightguru.com and request a free demo.
Thank you, Laura. We've just started our partnership with Greenlight Guru and have really enjoyed it. So I'm grateful for this opportunity to speak to our mutually interested clients. So I just want to give a background of myself first. So as Laura mentioned, I have worked in medical device for 25 years. I actually started in bioburden, doing radiation studies, radiation validation studies, and that included, microbial IDs and spending the time in the clean room. And I really decided I am not a microbiologist. I do not find joy in it. And so I was given the opportunity to move over to our biocompatibility department to bring up the in vitro chromosome aberration assay, which is one of the genotoxicity assays that can be performed. And I really enjoyed this study and spent the next 15 years in the laboratory doing all of the testing that we offer at Nelson Laboratories, as well as working with our partnerships with animal labs for the in vivo testing. And then just about 5 years ago, I started doing some consulting work and realized that I would really like the opportunity to work with our clients directly a little bit more and with the regulatory agencies and provide that mediation that's needed from the data to the regulatory agencies. So I've enjoyed my time in the consulting team. So we're going to jump right in and talk about the approach to a biological safety evaluation. One thing I want to just put right up front is there are many different types of submissions for agencies around the globe. But one thing I've noticed is consistent is that the requirement for biocompatibility, regardless of the submission type, is the same upfront. So whether you're doing for a clinical trial or for an investigation, the agencies really want to know upfront that the device is safe for patient exposure. And so keep that in mind as you think about this and you're thinking about your own types of submissions, that these considerations apply no matter the type of submission. And sometimes there's time leeway that we can gain, but not really, justifying out of a whole lot of addressing some of the main concerns. So we'll talk about that as we go through the presentation today. So the approach that's laid out in the standards really goes through a 3-phase process. So this is short and sweet on a screen, but we all know biocompatibility can take some time. But we just want to talk about it in these 3 general categories. So the first step is to develop a biological evaluation plan. This is where we really talk about the device and get to know the device, its materials, and exactly how it contacts the patient. And then we step into the testing and risk assessment. So phase 2 is really carrying out everything that you put in your biological evaluation plan. And then phase 3 is a biological evaluation report. So this is really your chance to say, did everything go as planned? What were the results? And has somebody with the right experience looked at everything to say, okay, our device at the end of this has shown that it's biocompatible and it does meet the standards. So we're going to keep visiting this slide as we go through the presentation today. And as we go through these 3 phases, some key points that I want to touch on are specifically how to meet the physical and chemical information requirements. I see a lot of companies get hung up on this initial part and even regulatory agencies. So I like to have the opportunities to present a thoughtful approach to this requirement. Also, when justifications are appropriate, so when you want to justify maybe doing a different test or you want to justify not doing testing at all. Also, considerations that should, considerations when you're submitting samples to the test laboratories. So things for medical device manufacturers to keep in mind. And then also addressing failed tests. So when we think about biocompatibility, I automatically think of ISO 10993. So this is a series of standards that addresses biocompatibility and how we approach that. And ISO 10993 is all about risk. So the title says, evaluation and testing within a risk management process. So we need to think about what the risk of the materials and processing of the device are to the patient. And that's really what we're assessing. And so I like to pull a definition of risk from ISO 14971, which is the document that helps us know how to write risk assessments. And this document states that risk is the combination of the probability of occurrence of harm and the severity of that harm. So how likely is the harm to occur? And when it does happen, how bad will it be? Are we talking a rash or are we talking death? So we want to keep those 2 things in mind. Another tool that we use is in ISO 10993 Part 1, and this is Table A1. If you've not heard reference to this table, you will repeatedly. This is the go-to check, I guess I would say chart that we want to check for every device that we evaluate. So I just want to point out how we use the table here. And we first always start with how does the device contact the patient? You'll hear me talk about this a lot because I recognize in a lot of submissions that I'll review or that I jump in on to help discussions with regulatory agencies, that there's a lot of confusion because not enough time was taken in this initial discussion. So just think about, this is how we want to think about biocompatibility. So we talk first about our category and then contact. So is it a surface device? Is it contacting skin or is it going to be internal with the circulating blood? We also have implants. There's a second half to this chart. And then we talk about the contact duration. So less than 24 hours, 1 day to 30 days, and then greater than 30 days. And then you'll see that there's marks in each of these endpoints. So we've kind of taken the human body and broke it up into endpoints that we can evaluate. And we've marked them with an X or an E. And I just want to highlight here, so this is the second half of the table. And we get the definition for X and E here. So X means it is a prerequisite information needed for your risk assessment. And then I'm going to go back here. You'll see that the only endpoint required is physical and or chemical information. So we have to gather this information before we can move forward with deciding what tests are appropriate and which tests do we not need to perform. And then E is for evaluate. So I've put this on here for your reference for after the fact, but this is straight from the standard. When we think about evaluate, that doesn't mean you have to run out and do all of the testing, but it does mean you need to present data for that endpoint. So if you have all three, like cytotoxicity, sensitization, and irritation marked for your device, that's required, then we need to present data and proof that the device is not cytotoxic, a sensitizer, or an irritant. And we can do that in many ways, and we'll talk about some options for that as well. Okay, back to this slide where we break it up into the three parts. We're going to dive into a biological evaluation plan and talk about some details there first. So this BEP is really a critical first step in biocompatibility. I do see a lot of responses from submissions that say, there's no risk assessment that says why you did the testing you did, why this is supposed to support the safety of the device. So agencies really do expect proper documentation, and a biological evaluation plan is not only great for your submission, but it's great for a company. It's a way to capture tribal knowledge to say why you did a hemolysis testing or why you didn't do implantation testing. You don't want that to leave with employees, right, when they leave the company. So I recommend this even if you don't have to submit. Maybe it's a low risk device and you just need to put this in your device history file. I really recommend having this initial BEP. And just some requirements from ISO 10993 Part 1. It does say that your biological evaluation should be a structured process and should be part of a risk management process, so should be risk-based, and that this should be carried out by the appropriate personnel who have the right credentials and experience to carry it out. So I have had to submit my CV several times for clients that we've worked with. So I would just encourage, if you have staff members that are your biocompatibility experts, to really be thoughtful of, are they supported and will their CV support our biocompatibility submissions? Okay, so ISO 10993 also offers this chart. So the table, which we'll show again here, but this flow chart to say, how should you structure your biocompatibility? And the very first question is, does the device touch the patient? And you'd be surprised how many times I actually talk to clients and say, there's no contact with the patient here. We don't have to do this testing. So really think about direct and indirect contact. But the second portion is this requirement: obtain device material and consider material characterization. So I just want to point out that it doesn't say you have to do material characterization testing. It just says you need to consider if it's necessary. We're going to get back to that and break this out into some thought processes here. And again, here's our table showing us that physical and our chemical information is the only required point and is our first one we're going to step into. So how do we present this physical and our chemical information? What is the actual requirement? So 10993 Part 1 actually gives us a definition for physical and chemical information. So the definition tells us that we're just gathering and presenting our knowledge regarding the formulation of our product, the processes, any geometry and physical properties that are important to the function of our device, and then the type of body contact and the clinical use. And this is an important feature because we'll go through that discussion and then say, there were some compounds here that we're concerned about. We are going to need to do chemistry testing, or because this is contacting the patient for more than 30 days, chemistry testing makes sense. And I'll go into some more details on how we get to those decisions. I also want to define or put up this definition for material characterization, that this is more in depth. So this would be the testing. So you're still collecting that information on the material chemistry, but you're doing this now through some chemistry testing. But this is not the initial requirement. We have to determine if this is the appropriate process. Just like we don't do all of the animal testing, we have to decide which ones are most appropriate. That is the requirement here as well. So some options to meet the physical and or chemical information requirement. We can get supplier information, predicate testing. Maybe this is generation 3 of your device and you've just added a longer tube or a longer catheter, or you need to do some material or chemical characterization testing. Material characterization and chemical characterization are interchangeable terms that we use here. I find in the standards they're mixed as well. So if I say material characterization, it is the same in this conversation as chemical characterization. So let's talk a minute about supplier information. I love to get information on products or materials that are already claimed to be biocompatible. A lot of times I'll see things like ISO 10993 compliance or USP class 6. It does tend to make me ask more questions to the supplier, which I typically can get those answers for. So I know, they're cooperating pretty well. When we say ISO 10993, I don't exactly know what that means because there's over 20 standards and multiple tests in each standard. So I like to ask clarification for which standards were followed or which tests were performed. And then I like to understand the form of the product that was tested. Did it have the same colorant that I'm using? Was it sterilized through the same process? Do I do any different manufacturing processes to the material that would make those test results then not really, not directly correlate to my device? And with USP Class 6, I asked those same questions as far as what form was the material tested in. USP Class 6, I already know what testing that is because it's laid out in the USP. It includes an irritation and acute systemic toxicity and an implant test. But you'll notice that USP Class 6, it doesn't really cover any full ISO 10993 endpoint requirement, but it can help you get to where you need to be, and it can reduce the testing plan if the testing does correlate directly back to your final device. Predicate testing, I just put this quote up here from the US FDA just as an example that, the FDA says if you do predicate testing, you have to provide a specific comparison table, and they like a side-by-side. This is, the device that was tested, and here's our current device, here's what's different, and then a justification for why the testing applied. So I just put that up there to highlight that the agencies are open to that, to using comparison data to support the safety of its subject device. Okay, and then the last option, of course, is to perform the chemical characterization testing. So the standard we use for chemical characterization is ISO 10993-18. And in the introduction of that standard, it specifically states that before you think about doing this testing, you really need to think about the nature and duration of your clinical exposure. And we're going to use that information to help us understand if this data is appropriate for the device. So with chemical characterization, we step into extractables and leachables testing as outlined in 10993 Part 18. We want to think about what does that data tell you, and then how can the data be used to support the safety of the device and also to reduce animal testing. So it's twofold with the chemistry testing. So what does the data tell you? So if you get a chemistry report, it's a list of compounds that were identified and how much came off the device. Then from there, you do have to step into a toxicological risk assessment. So chemical characterization testing is a twofold process. You do the extractable leachables or ENL testing, and then you have the data evaluated by a toxicologist. So the toxicologist can assess the data to understand a systemic exposure to the patient. And this is based on the type of literature that they can find and also based on thresholds that are established, knowing that we have certain thresholds for carcinogenic compounds or systemic toxic compounds, but there's some endpoints we don't have those thresholds established quite yet. So just for an explanation, systemic exposure that we have identified thresholds for that we can use chemistry for is acute, subacute, subchronic, and chronic toxicity, genotoxicity, and carcinogenicity. And so you think, like, I know a lot of places like to use sensitization, and I do think that that is on the verge, but we don't have widely agreed upon thresholds for sensitization. And so the agencies aren't widely accepting that. So just something to keep in mind. Okay, so I went back to the tables and I just highlighted for the type of device contact that would be most applicable to use this chemistry testing. And so you can see I put a star next to them, and I starred the ones that are prolonged and long-term, because if you look at the endpoints that need to be evaluated, this covers a lot of systemic toxicity testing. So I've marked that through here. So just some things for thought on that point as well. So this is a very, very general approach to chemical, to meet this physical and or chemical information requirement. So if you have a limited contact device, our first thought approach is to identify the materials and processing. And if there are no compounds of great concern for the type of contact with the device, we recommend to use the biocompatibility testing to support safety. If you have a device with prolonged contact, again, you'll identify the materials and processing. You'll use your biocompatibility data, and then consider if chemical characterization is appropriate, and that may save you some animal testing and some funds. So in the prolonged, it's kind of a gray area depending on the device contact and the material information that we gather. If you have a permanent contact or long-term contact device, then we do recommend to perform chemical characterization testing, and that has been the standard requirement from regulatory agencies. So again, just a general thought process. Of course, there are different caveats for every single device, but this is where we generally start and aim for as we look at devices. Okay, we're still talking about biological evaluation plans, and here's some considerations when you think about and prepare for submitting samples for testing. So the question is, how do you pick what you should test? Again, do we look at just the raw material, which we have polypropylene pellets here on the left, or do we need to include our finished device? And again, we want to include all the processes. So biocompatibility is an evaluation of materials and processes. And 10993 Part 1 does state that the testing shall be, so that shall is a command, right? We have to follow it. It is a requirement. It shall be on the final finished device or a representative sample. So I just wanted to talk quickly about representative samples. You can use coupons, so something that represents your sample. If you have small or expensive devices, typically we don't want to include 1,000 devices in an extract if the patient's exposed to 2, right? So we want to think about patient exposure and consider justification of maybe a different extraction ratio. And with small, expensive devices, we'll also consider the use of coupons. If you have a product family, you could pick one product that represents all the materials and processing for your family, or you can make a master product. We also call it like a Frankenstein, where you may put together a few, different connectors or something to make your monster product that we would say represents your full device. Okay, another thing to think about for test sample selection. So this is not really, picking a representative sample. This is like thinking about how you'll need to break up your testing. So one thing we come into a lot is multiple components in a system. It's rare if you have just one product that you're submitting. It's usually part of a full system. there's a good chance those products will need to be tested separately. So when you plan out your biocompatibility, if you have like a delivery device and an implant, you will most likely need to test those separate. And my thought process on this is if your delivery device is quite large and your implant is very small, we want to test those separately so we don't dilute out any effect that the implant could be having by testing it with this large, delivery device. So that's a typical consideration that we make in our recommendations. Another thing to think about is if you have a device with direct blood contact, or blood contact in general, if it's indirect, that requires a lot less hemocompatibility testing. If you have some that's indirect and direct, you may have to separate those components out for different hemocompatibility tests. So again, testing some things separately. And then even if you have some devices, especially with circulating blood contact, if they're used together, often our in vitro analyses make it very difficult, and even the in vivo analyses make it very difficult for us to assess those combined. So I'm thinking about like a dilator with a catheter that will be used together. Often we recommend to test those separate, and this comes from recommendations at the animal laboratory that they see a lot of issues because we have to run those tests under worst case conditions, not necessarily clinical conditions. So we do often see some adverse responses just based on the test system. So we do often recommend some separated, different separate testing for those components. And so just things that you'll be asked about and that you need to think about up front in your initial biological evaluation plan. So the next thing in your test sample preparation is you'll be asked questions by the laboratory, how do you want us to test this? And you may say, well, the lab, you guys are the experts, but honestly, the manufacturer is the central point of control for dispersion of testing. And so it is the manufacturer's responsibility to understand how their device is being tested so they can coordinate that to be the same across all of their testing. We can certainly help write this in a BEP, and it's great to put it in your BEP, but just wanted to explain why it needs to be kept that, those decisions with the manufacturer. So I wanted to give an example. This is a knee replacement device. And so, we have a couple different options for how we're going to test this. We could do by weight or by surface area. And this is, to determine extraction volume that we have to do for every biocompatibility test that we perform. If we do it by weight, this weighs just under 94 grams. So we're going to add, just about 470 mils of extraction fluid. If we did it by surface area though, it's quite a small device and we would only have 37 mils of extraction fluid. If we think about the difference here, the volume based on weight is 470, the volume based on surface area, 37. It's about a 12-fold dilution. And I think everything I've seen will pass at a 12-fold dilution when we've done that before. So when we run dilutions. So it's really important that we use the appropriate weight or surface area when we're extracting the device, because we want to create a concentrated system, or a concentrated extract. And sometimes weight is the better option, but like I think a thread or fabric weight or thread particularly can be better by weight. So we just want to be thinking about that, and so you have a justified approach on that extraction. Another thing to think about is, what extraction time and temperature. The lab will also ask you this. And I would say the most typical, so 37 degrees for 24 hours is only allowed for cytotoxicity, because 37 degrees is the highest temperature we can use with the extraction media. However, it has turned out to be that the FDA, and now it's included in the 10993 Part 5 and 12 standards, that for cytotoxicity, 72 hours is actually preferred if you have a prolonged or long-term exposure device, and that 24 hours can be used if you have a limited. But for every other test, the preference is to use the 50 degrees for 72 And when I say every other test, I mean mostly outside of hemocompatibility, because they have their own special extraction conditions that we comply to. So remember that when you extract, the goal is to perform an exaggerated extraction in time and temperature. So keep that in mind. And I would say, if you say, oh, my device is only used at body temperature and it's only used, for a day, I would say always start with 50 degrees, even if you're nervous that you'll have issues, because FDA and regulatory agencies will want to see data for why you have to use lower than 50 degrees for everything outside of cytotoxicity. And this is just for informational purposes, that there are some additional considerations for your extractions. You want to typically do in a polar and a non-polar. In cytotoxicity, we do use cell culture media with serum, and because that's compatible with the cell system. For every other animal test, we use a polar or a non-polar solvent, and those are listed here. And so I just put together a summary for what would be included in a biological evaluation plan. This is our typical flow, where we start first by describing the device, and then talking about the physical and our chemical information. And our device description plus the physical and our chemical information then lead us right into the appropriate recommended testing. And then we go through and discuss those special test sample requirements that we talked about. So now that we've kind of breezed through the biological evaluation plan, we want to talk about the testing and risk assessments. So again, stepping back into this X versus E on the table. So X means required, so you have to present your physical and or chemical information. And then E is to evaluate. And so you can evaluate it through existing data, additional endpoint specific testing. Maybe you've done a large animal study and you could wrap up some other endpoints in there, or additional rationale for why the testing isn't appropriate, but have the conversation about it. So I gave some options here for E, like maybe the data is already available. Maybe you purchase a component off the shelf and the testing's already done, or it's already like a 510K or CE marked device. And the material is well known, like you have a cotton fabric medical garment, or it's a titanium alloy and it's an implant. We have a lot of great clinical history of use information for titanium. Maybe the test does not correlate to the use of the device, like an implantation test for dentures. If you look at the type of contact of dentures, it would say implantation should be considered. So we just have a conversation in the BEP about implantation and whether it really applies. And then other options for E, again, using chemistry testing to address toxicity endpoints. So you could use your extractable leachables data and a toxicological risk assessment for a suture anchor, and that could address several of your systemic toxicity endpoints. So again, don't do all the testing necessarily. We just need to talk about all the endpoints and determine if the testing is appropriate. And failed test results, as much as we don't want them to happen, they of course do. And I would just say, if you have failed cytotoxicity, you are not alone. That is, it's such a sensitive test system, and it really isn't compatible with a lot of medical devices. So we do see a lot of failures. I put some examples here. If you have silver or copper or zinc, anything that's antimicrobial, it's antimicrobial because it kills cells. So it is cytotoxic and it will kill the cells in the culture. If you have latex, that's our positive control for the assay. So it definitely will kill the cells. And things like toothpaste or gels, they just mess up the osmolality and the balance of the solutions, and the cells really can't handle it. So we do see a lot of failures in med device with cytotoxicity. So things to think about, is there a comparator device on the market that you can run side by side and say, yeah, our toothpaste is cytotoxic, but so is Colgate, or I don't want to throw them under the bus, but any toothpaste will fail. So, you can show that it's just as cytotoxic and has been okay for human exposure. Maybe you can run dilutions to say, yes, it's toxic, but once we dilute out, if maybe you've determined what the cytotoxic effect is, you dilute that out and show the rest of the system is okay. Maybe there's an undetected change in your process. We had a company add an additional IPA rinse because they wanted their device cleaner. And even though we think IPA evaporates, enough of it sticks around that they started to fail their routine cytotoxicity monitoring tests. And we had another case where somebody said, hey, everybody wear gloves because you're touching the product. We want it to be clean. And they started to fail their cyto because the latex powder was coming off the gloves onto the product. So, you know, just some things to think about how to troubleshoot and think about that. Maybe you say, oh, nothing's changed in our process. Any change is a change, right? Even if you think, oh, it's making it better, it could be impacting things down the road. With the IPA rinse, we just said rinse with water after that. And they did, and it was fine to get the rest of the IPA off. So just some additional food for thought. So when we go to our last point, this biological evaluation report, again, this is the summary report where we talk about everything that happened during phase 2 and if it really matched up with phase 1. And so kind of a similar flow and discussion as a BEP, but I will say that there's a few extra discussions in there, like if you're submitting to the EU, we talk about carcinogens, mutagens, reproductive toxins, endocrine disruptors. That's part of their GSPR 10.4.1, I think, in the NDRs. We talk about the test performed, we talk about any failures, we can talk about clinical history. So maybe this BER is on, you know, again, a generation 3 or something of the device, and we already know that, hey, you know, we only changed X and we know the device is safe for patient exposure. So this was really the meaty conversation, putting it all together, and then hopefully coming to the conclusion that based on the test results, the information in this report, that the device is biocompatible and meets the standard requirements. So I know I really breezed through that. I told you guys that I could, you know, I typically talk about 8 hours on average about this stuff because there's so much to go over, but we can definitely help you out. We do offer all of these, you know, BEP, BER, any risk assessments. We do have board certified toxicologists to help you with your chemistry. And we definitely can help with regulatory submissions, having those conversations with your regulatory agencies. So if you guys have additional questions, I'd love to answer them now. And then also you can reach out through this, through our website, and also email me as well. So Laura, I will turn it over to you.
Sounds wonderful. Thank you so much. I can say I've certainly learned a lot from your presentation. I'm sure many of our participants that we had join us today have as well. So we have had a lot of wonderful questions come in as well. We're not going to have time to get to all of them, but we will certainly try and get through as many as we can here within our last couple minutes. That sounds great to you. I would go ahead and jump us in with our first question. Perfect. So it is that they're saying that my device is a mask, low risk material in contact. Do I need to do all of the testing and assessments, for example, a BEP or a BER, sorry.
Right. So yeah, masks and those protective equipment have been a hot topic. So on the fast track, when it was the emergency usage, a lot of the documentation was not required, but the testing was. And so, but now as companies are trying to get it officially approved, the documentation has been required. And I would say masks present a unique problem in that they typically fail cytotoxicity. Because the elastic on the ear loops is cytotoxic because it's latex or an elastomer. So in that case, consult with the lab and test the ear loops separate from the face mask because it needs to be a separate data consideration.
That is very interesting. I had never thought about that with the ear loops part.
Yep.
Perfect. Well, thank you so much. I will continue us along in our questions here. So our second question that came in is asking, so my company recently purchased a device from another company. There is very little information, none to very little information on biocompatibility. Do we need to perform all the tests even though the device has been on the market for more than 10 years?
This is a great question, and a lot of the answer will depend on the company's comfort with regulatory risk. And so we know under the MDR that they really don't want to accept previous clinical data. They want to see that you have biocompatibility data to support the device. So it depends on the class of device, how risky the contact is. If we're talking an implant and there's not a lot of data, you're going to have to gather some data. So the recommendation there is to start with a biological evaluation plan, talk about what you have and work with the experts and consider where you are marketing the device, and then consider the recommendations for testing there.
Absolutely. Thank you so much. So our next question that came in here, it kind of goes along with what we were just talking about with the mask a minute ago, but it's saying in the presentation, you talk about us needing to tell the lab how to test the device. Why doesn't the lab decide on the preparation and extraction steps?
Right, exactly. And it is, you know, we can certainly consult with that and give you our recommendations, but the ultimate decision needs to lie with the central point. We understand not everybody is testing with Nelson Labs. We'd love for everyone to go through us, but we know you've got to get product tested and out. And sometimes that means working with multiple partners. And so since we are not aware of all of that testing, you really, as the manufacturer and the coordinator of that testing, do have to be aware of the testing parameters and be involved in that so that you can communicate that to all of your partners.
Absolutely. Absolutely. Thank you so much. Our next question that came in here is saying, for biocompatibility requirements, do they need to consider only the patient contact, or would you also need to consider the user contact?
Right, so 10993 Part 1 does talk about practitioner contact, that we need to be protective to the practitioners. What I find most of the time is that the practitioners are wearing gloves. And when that's not the case, then we do have a conversation about any potential risk to the practitioner. But typically it's skin contact, it's low risk. So, and usually if there's a liquid or a chemical, I do find that the requirement is to wear gloves. But we do mention that briefly in a BEP to just show, hey, we've thought about it. This is the intended contact, and here's, you know, the risk level that's there, and whether we need to address that further or not.
Absolutely. Perfect. Thank you so much again. We've got a couple more minutes. I think we can squeeze in one or two more questions here. So one that has come in is asking, what would you recommend doing if your test came up with unidentifiable compounds that are above the TTC? How would you approach going about that as far as your next steps?
Yeah, so this is something that our toxicologists deal with a lot. And I would say your best bet is to see and hopefully get a toxicologist who's also a chemist. And that's proven to be very beneficial because they can look at those unidentified compounds and then determine, you know, thinking about the contact with the patient population, and then to determine how could we improve the method. Could we increase the number of samples so that we can raise that unidentified compound? Maybe we just need to make the test more sensitive. And then also, I know that one of our toxicologists who is a chemist, or who was in a previous life before she came to Nelson, she has designed and helped to design targeted chemistry testing for a class of compounds for some of these unidentified compounds that came up. And so there's a wide possibility on how to approach that, but really partner with your toxicologist and your chemistry team to you know, do some maybe more targeted analysis, especially, you know, if you have like a neonatal patient population, you're going to have to get those identified. It's really critical.
Absolutely. That sounds like some wonderful advice. Thank you very much. So I know we are coming down to our last minute here. We have had many more wonderful questions come in that I know will be sent over to you, Audrey, at the end of our presentation. And you've also provided, it looks like, a website and some contact information as well for anybody that needs some additional help in our areas. But I would like to say a big thank you from myself, Audrey, and from all of our other participants for today for the wonderful presentation. So thank you very much.
Thank you for having me. I appreciate the time and the opportunity to talk about it. I could do it again for days.
Absolutely. I can say this is bringing us to the end of our second day here when it comes to our summit series. There are a lot more presentations to come in the next couple days as well. And hopefully in a future summit, Audrey, we'll get to bring you back again.
I'd welcome it. Thank you.
Perfect. Thank you, everyone, and have a great rest of your day.